able to answer live can be answered after the speaker finishes. Okay, next slide. So as you know, these programs are extensive research experiences that are funded by the National Institutes of Health. And the goal is to educate and immerse students such as yourselves deep into research, because we need to diversify our workforce. And so this is the goal of our programs. And we established this partnership with Dr. Tilak Ratnanathan a few years back. So I omitted him yesterday, but I was so delighted. The students did not omit you, Tilak, so they understand and know all of the energy and effort that you have put into this program. And so we're in our 3rd year now, and so it's really fantastic. And we wanted to share this video. Go ahead, Sarah. Johns Hopkins Neuroscience Scholars Program is for undergraduate students. This is a program targeting individuals who are underrepresented. This includes racial and ethnic minorities, as well as people who are deaf, hard of hearing.
I'm definitely excited to have the opportunity to be here at Johns Hopkins, just because it's a world-class university.
I've never had an opportunity to be around so many other deaf and hard of hearing people before, so that's a very new experience for me, which this program is doing a great job of getting people like us involved in research.
It's actually like reaffirming that, you know, I'm okay and that I can actually be smart and dream like big.
I also feel like just in the growing age, we're just trying to have more diverse people in the medical field and in the STEM field, just because I think like diverse minds and different backgrounds can contribute to better things.
Once in a lifetime experiences, they don't come around very often, and this was one of those opportunities for me.
These are students who are curious, they're motivated, they want to make a difference, they want to be leaders. Today is a showcase, so the students, they actually get to now come out and practice their science communication skills.
The group that I'm working with in my lab has mostly graduate students. There's a few postdoc fellows. Everybody collaborates with each other. That's the great thing about it. It's all a very close-knit effort.
Overall, it's been a really learning experience, and it has opened my eyes to like even a career in research, as opposed to like everybody comes in here and talks about they want to go to med school and do certain things. It has like really navigated my path into a new direction that I wouldn't have like gotten anywhere else.
What we're doing here is really enhancing their identities as scientists, their self-efficacy as scientists, because they can now see the pathway forward. And with that is the professional development.
This program is basically a way of saying that I'm important. For so long, I've had to show people that I am important too. And this program shows that it's worth investing time and money into us, because we can be just as successful as other people.
Diversity and inclusion is important. It brings different stories, it brings different ways of thinking and approaching things.
This training that they are getting is going to set them up to be the next generation of leaders in neuroscience. That was so comforting to see again. I hadn't looked at that in years. So hope you, I don't know how many scholars had seen that video. How about those inside here? Okay, great. Awesome. Thank you so much, Heather and Sarah and the organizing team for remembering that. Excellent. Good. Well, it's my pleasure. I think we'll go ahead and get started. And so our first speaker today is Jennifer Hinton. Jennifer is a senior, right, at the university, Mississippi State University, and she's one of our alumni. And so I'm very excited to hear progress in her research from Dr. Burgos' lab. So Jennifer, take it away.
All right. Let's see. Can you guys see everything? Okay, great. Let's start. All right. So here we go. Hi everyone. Today I'm going to tell you about my work in finding expression of prunergic receptors in the developing cochlea with single-cell RNA sequencing. For a little background on the cochlea, sound happens when sound waves hit the eardrum, and those vibrations travel through the bones in the inner ear. Once it hits the third bone, those vibrations will then send signals through the fluid chambers of the inner ear like the scala media. These signals will then induce pressure differences that cause the basilar membrane to deflect, which in turn will move the stereocilia, those little hair-like cells in the picture, and those are on hair cells. This movement will open up the gated ion channels that allow the hair cells to depolarize, release neurotransmitters, and activate the auditory nerve. Another important note to tell you is that the supporting cells surrounding the hair cells on the right, on both sides, all contribute to the hearing function by giving structural and metabolic support. So one thing that is important in hearing function is the extracellular signaling molecule called ATP, which is known to activate both classes of prunergic receptors called P2X and P2Y. The main difference here is that the P2X receptors act as an ion channel for the diffusion of cations such as potassium, calcium, and sodium, and P2Y receptors will act as a decoupled protein. Both of these are activated by different agonists. So why do we care about P2X receptors? All of the P2X subtypes are activated by ATP, which is released for protection when dangerous noise exposure happens. Also, literature has shown us that variants or mutations in the P2X2 gene have caused noise-induced and sensorineural hearing loss in human cases. P2Y receptors are also important for a few reasons. One, they are activated by various agonists such as ATP, ADP, UTP, and UDP. Secondly, the P2Y1 subtype has been found to be essential for spontaneous activity, and when the same receptor is blocked, all the spontaneous activity in the supporting cell stops in the first week and a half after birth. Spontaneous activity in the developing cochlea is similar to the way tinnitus operates in humans, which is the ringing of the ears. The supporting cells will spontaneously activate themselves and consequently stimulate hair cells, which in turn activate the auditory nerve when there's nothing actually activating in real life, such as sound waves. So these random bursts of signals at random frequencies are a way for the inner ear to kind of test and train itself for when sound actually needs to be heard after development. So some problems in studying prunergic receptors right now are that we don't actually understand the mechanisms in development, and the literature currently shows low expression in the developing cochlea. So when we try to find the expression ourselves, it can be quite difficult because the antibodies and immunostaining can be difficult and inaccurate because they don't stain well. And so this can give us, as a result, inaccurate results. So to solve this, we try to use single-cell RNA sequencing. To do single-cell RNA sequencing, we focused on postnatal day 7, and this took RNA from over 30,000 cells in the cochlear sensory epithelium. When looking at the data, it shows thousands upon thousands of rows of gene names, as you see on the left, and it shows the nucleotide bases on the column in the right, and you can see the numerical level of expression. And so it would take us forever and ever to look at all this data row by row, and we use a great technique called dimensionality reduction to get past this. And what it does, it takes all of this high dimension data and put it in a low dimension to let us analyze it. And so as you see in the TSNE plot, it shows clusters of organization based on things that are similar. So clusters that are further away from each other, like the ones labeled 8, 9, and 11, show more things that are different, and then ones that are closer together show more similarity in their genes, such as the ones labeled 1, 2, 0, and 4. And it's worth noting here that each dot on this plot that you see represents 3 extracted cells from the RNA sequencing. Another thing that we have to do is figure out what cell type the clusters represent, because when we see the data, it doesn't tell us what cluster the RNA was extracted from. So for each cluster, we take the top 5 differentially expressed genes, and we use these genes to isolate expression to one cluster, and then use immunostaining from literature to determine the cell type. So here I have one example with outer hair cells. The purple represents expression levels, and the only purple shows up on that one cluster labeled OHC, which is outer hair cells. So to verify my work, we looked at immunostaining that shows Preston, which is encoded by the SLC26A5 gene. Preston only shows in the membranes of outer hair cells and not inner hair cells, which verifies my work. Now we branch into P2X2 expression, where we see widespread expression in cell types. The bulk of it shows in supporting cells like uterus and the LGER. And P2Y expression shows again in the LGER and spread through the middle clusters, which are all supporting cells. This validates the immunostaining done by a postdoc I have worked with the past 2 summers. Travis has shown that at P7 on the columns at the right here, this shows that the only ones that to express P2Y1 are shown by the purple beta-gal antibody. This antibody does not overlap over the hair cells shown in green by the CALB antibody. So once again, it verifies what I've done. So to compare patterns of expression for P2Y1 and P2X2, I created a feature map that combines the expression of both genes and overlaps them. And on the color scale to the right, the brightest green and red colors represent the highest expression for the individual genes. The yellow in the top right represents where both genes had high expression in the same exact place. So the only place these both overlap is in the LGER. And this tells us that the supporting cells of the LGER play a huge role in the developmental hearing function as well as spontaneous activity. So to conclude everything, we found P2X2 was widely expressed in the developing cochlea. P2Y1 had high expression in supporting cells known for spontaneous activity. And in the future, we can do in situ hybridization and calcium imaging on the LGR to give us more insight on how the mechanisms work that could actually help us understand tinnitus better. And later on, we will try to do sequencing RNA from nuclei of an adult mouse cochlea to examine how expression changes from the developing ages to maturity, because we know right now that the LGER cells morphologically change as they approach adult ages. So last, I would like to thank my postdoc Calvin Kurzbergen and Dwight Burgel for allowing me to come back for 3 years. And last but not least, I would like to thank Dr. Brown, Dr. Ratnanathan, and Ms. Thomas for allowing this opportunity to happen. Thank you for listening.
Good job, Jennifer. Beautiful images. And it's just wonderful to see the progress that you've made and then connecting that with the RNA sequencing data and your results. I apologize. I was a little bit back and forth. So I hope somebody has a question for Jennifer. You can go ahead and unmute yourself.
I guess I have a question. It's a little not completely formed because you're doing a lot of amazing research, but so you looked at these LGER cells in the developing cochlea. So I guess I was just wondering what research you know about for like LGER expression in adult mice.
We don't know a lot just because one, the problem with adult mice is that the cochlear bone is very hard to get past. And so it's very hard to either get immunostaining done because we have to go through such an extensive process to decalcify that bone. And we do know right now, I couldn't find a picture early enough in time, but the cells for the LGER kind of act as progenitor cells and that they change to form something different in the adult age. And that's why we really want to focus on that because we think that the patterns will change significantly.
Thank you so much, Sarah, because I think that's a common theme that's been coming up in several of the talks is development, the development of the nervous system, over time and where the lesions happen. But again, adding to the complexity of solving these disorders of the brain, of the nervous system. Thank you. Excellent job. Okay, next up is Ashley Yim. Ashley is a rising junior at the University of Maryland, College Park. And she's going to tell us what she's been doing it's kind of evolved. Ashley, take it away.
Okay, thank you. Sorry. Okay, hello, my name is Ashley Yim, and as Dr. Brown has previously mentioned, I am a rising junior at the University of Maryland, College Park. This summer, I had the opportunity to work under Dr. Amanda Brown, where I expanded my research from the previous year on osteopontin, or OPN, into CD44. So over the next few minutes, I will be going over CD44 expression and its relevance in the central nervous system, or CNS. Before I do so, I have nothing to disclose. So what is CD44? CD44 is a multifunctional transmembrane glycoprotein that is broadly expressed. While its role in the CNS is present, it is not widely covered. Some major functions that CD44 plays in our body are cellular adhesion, including clustering and migration, myelopoiesis, lymphopoiesis, release of cytokines, hyaluronic acid or HA metabolism, and the activation of lymphocytes, monocytes, and keratinocytes. Similarly to how many roles CD44 carries, this glycoprotein is also present in various isoforms. Through alternatively splicing, which is a process by which exons are either added or deleted from the pre-mRNA, CD44 is encoded by 20 exons. 10 of these 20 exons are constant in all isoforms. These are exons 1 through 5 on the N-terminal domain and 15 through 19 on the C-terminal domain. CD44S, or the standard CD44, is encoded with all of these 10 constant exons, and CD44V, or the variant isoforms, results from alternatively splicing with the 10 constant exons and any combination of the remaining 10 variant exons 5A through 14. Exon 6, or CD44V1, is lacking in humans. There are also post-translational modifications like glycosylation, phosphorylation, and cleavage that can affect which CD44 isoform is expressed. So like osteopontin, the definitive establishment of whether or not CD44 is pro or anti-inflammatory is blurred. As previously mentioned, CD44 plays a role in lymphocyte activation. In addition, it also affects lymphocyte maturation, homing, extravasation, and infiltration. CD44 has shown to have pro-inflammatory roles in cutaneous edema, arthritis, and colitis. But on the other hand, in CD44 knockout mice, the inflammatory cell migration and cytokine production of tumor necrosis factor alpha and interleukins 1 beta and 6 increased. Also, in CD44 deficient mice, lung inflammation was enhanced, mostly due to the increased accumulation of HA. My first central idea that I came across was how CD44 is expressed differently in the CNS. Due to the time constraint, I'll provide a very brief overview of the trends that were observed. In all regions that were examined, which were the basal ganglia, hippocampus, cerebellum, and spinal cord, there was no immunoreactivity for CD44V6. Standard CD44 was largely present on white matter astrocytes and had no immunoreactivity on or in neurons and axons. Variants 4 and 5 were localized in the cytoplasm of large neurons of gray matter throughout all regions observed. Conversely, variant 10 was found on large neurons and axons and endothelial cells in both white and gray matter. So it's important to note that the presence of the variant isoforms in neurons was seen to be localized in the cytoplasm, which delineates from the usual expression of CD44 molecules on the cell membrane or surface. As CD44 expression varies throughout the CNS, it also varies throughout the stages of a cell. CD44 seems to be more widely detected in precursor cells rather than in fixed adult cells. In the mouse brain, astrocyte precursor cells in the developing cerebellum, neural stem cells, and oligodendrocyte precursor cells at postnatal stages are positive for CD44 expression. In the developing cerebellum, CD44 was expressed in Purkinje and granule neurons, but was later only restricted to granule neurons in the adult cerebellum. Similarly, in rats, CD44 expression and developmental age correlate. So in the rat hippocampus and cortex at postnatal P10, CD44 expression peaks in pyramidal neurons. My next central idea will be about CD44 interactions, specifically to HA and OPN. HA is also known as the principal ligand for CD44. It is one of the most specific ligands for CD44 activation, and all CD44 isoforms contain an HA binding domain located at the N-terminal region of the extracellular domain. It plays a variety of physiological roles, such as maintaining tissue hydration and osmotic balance, cellular proliferation, adhesion, and migration. CD44 is one of the regulatory factors for HA and its activity. As previously mentioned, it binds on CD44's N-terminal, inducing conformational changes that allow for the binding of adapter proteins and cytoskeletal elements, which later then activates other signaling pathways such as RAS, MAPK, and PI3K, which all activate or produce pro-inflammatory factors. The interaction conducts some of the functions that I've described in my introduction, like activation, homing, and extravasation of lymphocytes into inflammatory sites. Ironically, CD44's interaction with this principal ligand accelerates HA degradation. Similarly to the trends we notice with CD44 in development, HA levels peak in the embryonic developmental stages and then decline with age. CD44 HA interactions in the context of nervous system development are crucial for several processes, some of which are neural stem cell proliferation and neural crest cell migration. In the adult CNS, CD44 HA interaction is vital in regulating stem cell proliferation and differentiation. The loss of CD44 results in dysfunction in the hippocampus and the spatial learning and memory capacities. In adult rats, the loss of HA and foot pads increase pain sensitivity notified in CD44 dependent pathways. So CD44 also has vital interactions with OPN, which I will cover in the following slides. As I continue my research into OPN, I came across interesting points made about its interactions with CD44. In a cancer environment, the pathway of T cell inhibition, including the downregulation of interferon regulatory factor 8, which is a molecular determinant of apoptotic resistance in tumor cells, terminates repression of OPN. Thus, OPN is now able to bind to CD44 and suppress interferon gamma production, which is a pro-inflammatory cytokine. Contrastingly, CD44 interaction with thrombin cleaved C-terminal OPN results in the inhibition of IL-10 secretion, which carries strong anti-inflammatory properties. The binding interaction between CD44 and OPN induces mesenchymal cell migration towards wounds and enhances CNS stem cell proliferation, including neural and progenitor cells, neuronal differentiation, and axonal growth. Back in the cancer condition in OPN-positive prostatic cancer cells, or PC3, there's increased surface expression of CD44S, variant 6 and variant 10 in comparison to PC3 cells with no OPN. So I would like to thank Dr. Amanda Brown for giving me this opportunity to continue research. And I would also like to thank Dr. Claire Carter and our program administrator, Heather, for always radiating such positive energy. And I would like to thank all the scholars for providing an environment where I felt encouraged and appreciated. So thank you everyone for listening. And now I will open up the floor to some questions.
Great job, Ashley. That was such a very comprehensive review of CD44. I've learned so much, and Elizabeth is shaking her head affirmatively as well, because as you see, people are like, wow, what else don't we know about osteopontin, right? There is so much. It's such a fascinating molecule. Are there any questions for Ashley? Go ahead and unmute yourself. Let's see here. We have about 5 minutes. I do want to wait just in case for Shaheen, if Some of his mentors are going to join, so I don't want them to miss the presentation. So Ashley, what will be the first experiment that you'd like to do next summer in the lab?
Well, so the first thing that I would like to do is I would like to see what I've been reading and writing about to happen in front of my eyes. Because I feel like then I'd be able to get a better holistic understanding of what's going on, because I would like understand all the techniques and the underlinings of what's occurring. But in terms of trying to take a step forward, I would like to see how the interaction of CD44 and OPN carries out in maybe some animal models, because we've been talking mostly in the context of like neuroinflammation, but as I previously covered, or like I recently learned that it also it influences like the pain sensitivity. So it's stemming into areas that I haven't really looked at before. So I think that would be really interesting to observe.
Thank you. Excellent response. I agree. Okay, we'll pause just for 3 minutes and we'll start at 12:20. Thank you. Go forward. Okay, we are back, and it's with pleasure to introduce our next speaker. That is Shaheen Ahmadi. Shaheen is one of our his first year in the program, in our third cohort, and he's going to be telling us about his work in Dr. Wu's lab. Shaheen, take it away.
Thank you. I'm going to share my screen, and please tell me if you guys can see the slide, because I have 2 screens here. Okay, perfect. So hi everyone, my name is Shaheen Ahmadi, and in my presentation, I'm mainly going to talk about the background for my research project at the Wu Lab. Our lab is, broadly speaking, interested in understanding the molecular and genetic basis of innate behaviors, but has a particular focus on sleep and circadian rhythms. As all of you know, we all live on a planet that rotates around its axis, and its rotation creates 24-hour cycles in the environment. In order to anticipate these cyclical changes in the environment, like changes in temperature and light, virtually all organisms have evolved an internal biological clock. Over the past few decades, researchers have been able to understand a lot about the circadian rhythm and our innate biological clock by studying it in model organisms like mice and flies. In fact, 3 circadian biologists won the Nobel Prize in Medicine in 2017 for unraveling the basic molecular mechanism underlying the circadian clock by doing genetic screens in fruit flies. Now, a lot of our physiology is controlled or under the influence of our endogenous biological clock. And by physiology, I mean things like blood pressure, muscle strength, body temperature, and arguably the most intriguing and notable one, sleep. In 2014, the Wu Lab published a paper reporting their discovery of a gene and a protein called wide awake. They had conducted a large scale genetic screen in flies and identified a fly mutant that had a delayed sleep onset. And they named this mutant and the corresponding protein wide awake because of its phenotypic resemblance to people with sleep onset insomnia, who are wide awake at night when everyone else is asleep. Now, what the lab discovered about wide awake, which I will refer to as wake from now on, was that its levels increase at night in arousal promoting clock neurons, and that wake upregulates inhibitory neurotransmitter receptors, specifically GABA-A receptors, near the wake-sleep transition in the early night. This increases the neuron's GABA sensitivity at night and thus suppresses their excitability. Logically, the loss of wake causes these arousal-promoting clock neurons to remain excitable at night, and consequently, the flies can't fall asleep easily. Now, in ongoing work, the Wu Lab identified a protein homologue of wake in mice called ANKFN1 and discovered that, similar to the flies at the cellular level, ANKFN1, or wake in mice, rhythmically suppresses the excitability of clock neurons by upregulating GABA-A receptors and increasing the GABA sensitivity of the neuron at night. What these findings overall indicated to us was that the basic function of wake is conserved between flies and mammals. Interestingly, the non-neuronal isoform of wake was found independently and was named banderola. And it was discovered that its function it functions in the asymmetric cell division by basically moving signaling complexes around at different stages of the cell division. The researchers who did this work, they also argued that the basic function of banderola, or wake, is conserved in evolution. Now, the fact that the structure and function of wake is conserved has made us very curious about the evolutionary history of this protein. Because, as you may know, if something is conserved through evolution, it means that it plays a vital role in ensuring reproduction and survival. And the longer something is conserved, the more integral and foundational its role is in the species. This summer, we've been able to find evidence of homology between wake in flies and a protein in cyanoflagellates. Cyanoflagellates are emerging model organisms in biology and have a fascinating evolutionary relationship to us. They are among the closest living unicellular relatives of metazoans, which include all organisms in the animal kingdom, meaning that our shared ancestry with cyanoflagellates dates back to about 900 million years ago. Now, a lot of the work that I did this summer to find evidence for homology was conducting blast searches and trying to find proteins in clinoflagellates and other species that had similar amino acid sequence to wake and banderola. Then I conducted protein sequence analyses using SMART, which is a database that is used to identify and analyze protein domains. And I aligned the sequences that I found with the original wake protein sequence to find these conserved domains and amino acids. Now, the protein that I was able to find has all the distinctive structural features of wake, which include an ankyrin repeat domain followed by its fibronectin type 3 domain. This finding is very exciting for us because it offers us the opportunity to gain new insights into the origins and the evolution of this protein and to further our understanding of its role in our biology. On broader terms, it has also made us wonder if clinoflagellates have circadian rhythms. Do they have rhythmic metabolic processes? Do they sleep? And answering any of these questions can greatly contribute to our understanding of our own developmental history. With that said, I would like to end my presentation with a quote by Peter Holland, professor of zoology at the University of Oxford, who said, "To understand how animals evolved, we must study clinoflagellates." I would like to thank Dr. Mark Wu, who I believe is here with us right now, and Elijah Blank for their valuable mentorship this summer. I didn't expect to learn so much working remotely in such a short period of time, and I have them to thank for that. I would also like to give my special thanks to Heather, Dr. Brown, and the Johns Hopkins Neuroscience Scholars Program for supporting me this summer and making this research experience possible. It's really been my great privilege to be part of this program and work with such an amazing group of people. With that, I'm happy to answer any questions you guys may have.
Awesome job. Yeah, we're just so excited to have you and have Dr. Wu's lab on board. Very interesting. I have a question, but I'll let it open it up to the scholars first. Does someone have a question?
I do.
Go ahead, Jennifer.
So Shaheen, when you were talking about circadian rhythm, I was interested, and I apologize if you said this and I didn't hear. Does gender play a role in the effect of circadian rhythm with like GABA or other hormones or anything like that?
Gender, is that what you asked?
Yeah. Does gender play an impact in it?
That I actually don't know. I don't think so. At least I haven't heard anything about it.
Okay, so maybe this could be related. So next summer, you're in the lab. What will be your steps to grow clinoflagellates and start your experiments? Because I don't know anything about what the experimental model is.
Well, now that we've found the structural evidence for homology, I think the next step for this particular project would be to analyze its function in clinoflagellates. So right now we have an idea that they play a role in organizing asymmetric cell division, and they play a role in circadian rhythms. So it would be kind of designing experience that assesses either one of these functions and see what we can find. Because right now we don't really know how it functions in clinoflagellates. We just found out that the protein exists.
Yes,
there.
My question was even more basic. What do they eat and how do they sleep? Like how are you gonna keep them happy while you do your experiments?
Mm-hmm. Oh, that I haven't looked into it exactly. Yes, yeah.
Okay, awesome. Excellent job. Thank you so much. Okay, and next up is Darius Carr. Darius is he's on? Okay, good. Darius, I know you've been waiting for at least 2 years for this opportunity And Dr. Schnarr has embedded you deeply. We haven't even seen you. So we're all very excited to hear what you've been working on this summer. Darius, take it away.
All right. Oh, give me 1 second. All right. So this summer, I had the privilege of working in the Schnarr lab in the Department of Pharmacology and Molecular Biology. And I do want to thank Dr. Schnarr, even though he isn't here right now, for advocating for me to get in the lab. I think these past 3 weeks that I've been here have truly been a blast. Everything is new and everything is fun and exciting. So to start off, I researched ganglioside synthesis in the Schnarr lab and how they interact with proteins. So many people don't know what gangliosides are. I didn't before I started working here. When a lot of people think of the surface of a cell, they think of the phospholipid bilayer. When in reality, and Dr. Schnarr will say the same thing, this diagram is kind of like looking at a forest without the trees. There aren't any gangliosides on this diagram. These are what gangliosides kind of look like. This is just a cartoon example. They stick out on the surface. They're not like regular proteins. They're involved in some of the communication and regulation between cells. And those shapes and colors are kind of important. I'll explain that a little bit later. And there's two portions on a ganglioside. There's a lipid portion and there's a glycan portion. And the glycan is what's really sticking out there and enabling the cell to interact with other cells. So what I wanted to do, what I did this summer was first, we needed to transfer this GD1B or GT1B ganglioside into GD1B. And the main difference between these two is that GD1B doesn't have a salicylic acid on it. And the reason why we couldn't just buy the GD1B is because it's a whole lot more expensive than GT1B. And so it's a whole lot easier to just remove the salicylic acid and just get GD1B. And we use sialidase S, just an enzyme that takes off that salicylic acid. And now we've got GD1B. And this is just a TLC to kind of just validate that we do have GD1B. The far right column is the GT1B sample, and in the middle reaction column, that dark bar is the GD1B. We use a really cool computer program. There are a lot of bars underneath that solid bar, and we were able to find out that that solid bar makes up about 90% of GD1B. So the conversion was pretty successful. It's 90% conversion rate. We could have added more sialidase, but because I was only able to stay here for just a little bit, we figured it was good enough to move on. And so now we wanted to create a probe. The problem with ganglioside research is we don't know what gangliosides interact with which proteins. And so this probe would be introduced to cells. We would inject them into cells, and then the cell would interact with other cells, and it would create a really strong bond that is permanent. And we would be able to see which gangliosides interact with which proteins, which is incredibly valuable. And so we needed to add this portion that's circled in red. It's just called tag A. And so just to save time, in order to add the tag A onto the GD1B, we went through these steps: oxidation, amination, and then the amide coupling. And these are the reaction conditions if you're interested. And so after those conditions, we're pretty certain that we have the GD1B side tag A. But we use a device called the MALDI. It's a really big machine, and it shoots a laser at a sample, and it's able to tell the molecular weight of that sample based off of what comes off of that. And it's not really exact, but the molecular weight of GD1B side tag A is 1924 or 1923 after being shot with the laser. And you can see in that second peak, I'm not sure if you can see my mouse, but right here, it's 1923, 1625, which is kind of the same thing, but we are confident that we do have our material and that we have converted GD1B successfully into GD1B side tag A. And so in conclusion, I sourced GD1B from the GT1B that we bought just because it was cheaper. And then I synthesized GD1B side tag A from the GD1B that we made. And I validated my results with the TLC and the MALDI graph. And if I were to continue on this summer, the next step would be to deliver the probe directly into the cells to see which proteins it interacts with. And yeah, that's it. Any questions?
Okay, and I'll open it up to the scholars first. Any questions for Darius? Okay, excellent job. So many different techniques, and you knew nothing about TLC and MALDI. You say it like you've been doing it for years.
No, everything was a new experience.
Congratulations. How long, in terms of your reading or what you've, in your interactions in the lab, how long do the gangliosides persist on cell surface? What is their turnover? How often are they attacked by I guess it would be sialidases or glycosidases that are in plasma, or depends on the organ or the microenvironment?
Yeah, I'm not exactly sure, but I'm pretty sure, and I may be wrong, don't take my word on this, but I'm pretty sure it's pretty touch and go. Cells come in, they communicate with other cells, and once they're done transferring that information, that data, they continue on and continue to transfer data to other cells. Yeah, and that's why the tag is so important. It's kind of like a freeze frame, and we're able to see exactly what protein the gangliosides interact with.
You read something about how these gangliosides are added to proteins. Did you read about that in terms of inside a cell? What pathway, what molecular pathway or cellular pathways used?
No, I didn't read a whole lot about that, no.
Okay. So you'll be taking a deeper dive, maybe through the school year or next summer, because the endoplasmic reticulum, right, and through the Golgi.
Yeah.
If I remember back from my graduate school days. But yeah, this is very advanced research that you're doing. Typically, as you probably saw in the lab, it's really like graduate student level research.
Yeah.
Okay, awesome. So excited for you. Excellent job, Darius.
Thank you.
Okay, we get a 10-minute break and we will come back at 1 o'clock. So we'll see you back here. Thank you. Hi, Tilak, how are you? Yes, I was talking to you, Dr. Ratnanathan. How you doing, Tilak? That's the Tilak.
To myself. I'm fine, thank you. Good. Yeah. I
Doing
think I'm
fine.
meeting Market tonight.
You're doing what tonight?
Meeting Market Milani.
Okay, okay, you and Marcus are meeting tonight. Beautiful, you're meeting with Tilak.
Oh, yes.
Okay, excellent. You see, because Marcus came, I said, because it was immediate. The barrier, I'm like, I have the solution. Clear mask. You see, I have to stay in practice. Okay, because we had this, these are the Tilak's former mentees, developed this. Yes, so you can take those. I have another box and we can buy more. So if Jennifer, Ben, maybe we should just include it in their box that we'll send you guys. Okay, awesome.
So she is gonna try log in here and she will be home today. She took up with Carr.
Awesome. Okay, great, excellent, thank you. Okay, and we are preparing. Rose, you all ready? Okay, excellent. Next up is Rose Broderick. She comes to us from Lafayette University. She's in our third cohort, and this is her first summer, and we are so delighted that she's able to be here with us in person. And so she will be presenting from here. And Sarah, you're going to control her slides, that's correct? Okay, great. So we can clap, so clap, okay? Then we're gonna clap.
Hi everyone. The slides are not up there. Perfect. Okay, hi everyone. My name is Rose Broderick and I had the honor of working under Dr. Marco Grados in his lab this summer. We were investigating the inflammatory disorders of asthma and obesity and how they relate to psychiatric disorders in youth. Next slide, please. Obesity is defined as a body mass index exceeding 30. The condition is known to promote chronic inflammation due to increased circulating amount of the pro-inflammatory hormone leptin. Leptin plays a role in regulating your appetite, so more circulating leptin leads to reduced appetite. Leptin also influences cytokine production and regulates the hypothalamic pituitary adrenal axis, or the HPA axis, as well as the endocrine system. And the HPA axis regulates the sympathetic nervous system, so dysregulation of this axis can lead to psychiatric problems. The condition of asthma is characterized by inflammation in the airways. Individuals with both asthma and obesity tend to respond less effectively to asthma treatment and experience higher disease burden. Asthma is also associated with anxiety disorders, with higher severity of asthma indicating an increased risk for developing an anxiety disorder. Lastly, associations exist between various psychiatric disorders and inflammation. To name a few: ADHD, anxiety disorders, OCD, PTSD, and mood disorders. Next slide, please. Actually, no, that's good, thank you. Our hypothesis is that there is an association between the inflammatory disorders of asthma and obesity and certain psychiatric disorders in inpatient youth. Now, next slide. So demographic and psychiatric data were collected from 688 psychiatric inpatient youth between the years of 2010 and 2014 for this study, including primary psychiatric diagnosis at discharge. That's one of the most important things here. Next slide. Predictors for the psychiatric diagnoses were as follows: 0 equals no asthma, no obesity, 1 equals asthma or obesity, and 2 equals asthma and obesity. In this specific analysis, there were 19 psychiatric diagnoses used, and a non-parametric trend test was applied to the data for this analysis. So significant trends were found for 5 of the psychiatric disorders, 4 of which were positive and 1 being negative. For the sake of being able to visualize visually demonstrate these trends, we graphed the means of psychiatric disorders. Otherwise, the differences in the number of individuals in group 0, 1, and 2 made the graphs really tricky to interpret. So that's why we did that. As you can see, oppositional defiant disorder and mood disorder not otherwise specified exhibit a positive trend. Next slide. Disruptive behavior disorder and ADHD also exhibit these positive trends. And next slide, please. Anxiety disorder, not otherwise specified, shows a decreasing trend in this case. To summarize, significant trends connect the inflammatory conditions of obesity and asthma with select psychiatric disorders in inpatient youth. In the future, it would be productive to investigate the nature of the relationships between these conditions. One pathway could be to investigate a common causal factor linking them, some factor increasing susceptibility to inflammation and thus increasing the risk for inflammation associated disorders. To recap on leptin, leptin is a pro-inflammatory hormone that regulates energy homeostasis, the endocrine system, and the HPA axis. Its broad regulatory roles and immune system involvement make it worthwhile to investigate in relation to inflammatory and psychiatric disorders. Another direction we can take in the future is looking into possible sex differences in the data set by splitting the data by male and female and performing the same NP trend test. That way we can see if there are any associations between sex and these disorders. Females are overrepresented in autoimmune conditions, making up approximately 80% of all those diagnosed. So it would be interesting to see how our data fits in with this. Overall, understanding the interaction of sex and immune system function may provide more insight into the mechanism behind inflammation and consequently the inflammatory and psychiatric disorders that we're studying. I would like to thank my mentor, Dr. Marco Grados, for letting me be the first person to present this information, as well as Jana Bukhari, Jessica Chang, the Grados Lab, the Johns Hopkins Neuroscience Scholars Program, Dr. Amanda Brown, Dr. Tilak, Heather Thomas, and Dr. Clara Carter, as well as all of my fellow scholars for supporting me this summer. I had a very nice time.
Excellent job, Rose. You can stay right there. And I know you did this in
1 week.
in 1 week. So Rose, can we have the lights up, please?
Yeah.
Yes. So Rose has been phenomenal, showing phenomenal resilience and persistence, and learned something about communication. And so I'm just so delighted to have her in the program, and look forward next summer to diving deeper into your study. Thank you. Any questions for Rose? Go ahead and unmute yourself. Okay, if not, thank you so much.
Thank you.
Great job. Just checking the chat. Okay, great. Excellent. So is Kirian on board? Kirian is here?
Yep, sharing my screen right now.
Okay, excellent. So and I just don't want to move around too much here on the screen. So it is an absolute pleasure to introduce Kirian Kawachi. Kirian is part of cohort 2 And so he's going to tell us what he's been researching this summer. Take it away, Kirian.
Thank you, Dr. Brown. And I would just like to first off say thank you to everyone who's here, taking out time out your day to, listen to all the scholars' wonderful presentations. But like Dr. Brown said, my name is Kirian Kawachi. I'm a junior at the University of Pittsburgh, and I'm cohort 2. I've been working with the Johns Hopkins Neuroscience Scholars Program for the last 2 years under the direction of Dr. Brown and her laboratory. And today I will be telling you a little bit about tracking neuronal responses in HIV-associated neurocognitive disorders through neuro-oscillations. Now, I just want to go briefly over some HIV-associated neurocognitive disorders and why it's important for the research that I'm doing. So, of course, HIV, human immunodeficiency virus, is a virus that's characterized by its attack on the immune system. And in 2020, about 37.7 million people across the globe were affected with HIV. And out of that 37.7 million people, about 27.5 million people are actively using antiretroviral therapies to combat the virus. Now, although antiretroviral therapies have greatly decreased mortality, HIV-associated neurocognitive disorders, otherwise known as HAND, remain prevalent, and they greatly decrease the quality of life for those affected. And so some deficits of HAND, HIV-associated neurocognitive disorders, include impairments to attention, motor processing, motor function, and perception. So understanding the neural basis of these deficits associated with HAND could end up leading to earlier and more efficient, more accurate diagnoses and more precise prognoses. So the million-dollar question, what exactly are neuro-oscillations? So to keep it simple, neuro-oscillations are repetitive or rhythmic neuroactivity in the central nervous system. So now when information is transferred at the synapse, extracellular currents are generated at the end of these dendrites of the receiving neuron. And so the neuronal currents that spread through the brain to the skull can then be monitored through the electrode. And so it's interesting because the mechanisms behind these oscillations vary. So for individual neurons, you can see neurooscillations generated due to the action potential or membrane potential. And then when you're looking at clusters of neurons, these interactions between neuronal groups, you'll see that synchronous activity ends up looking like this image that I have below, with the local field potential and the neuronal spiking. You can see that all these different neurons, these dots, each individual dot is supposed to be a neuron. And you can see that when they fire in clusters like these, it's very much correlated with the amplitude, the spike that you see in the local field potential there. So I also want to bring to light that neuro-oscillations are also characterized by frequency, amplitude, and phase. And in this image above here, titled Human Brain Oscillations, you can see different types: the delta, which is the 1 through 4 hertz, theta, which is 4 through 8 hertz, alpha, which is 8 to 13 hertz, beta, which is 13 to 32 hertz, gamma, which is 25 to 140 hertz. Now, I really want you guys to hone into the amplitude, because the amplitude in a variety of studies has been linked with synchronization. And the main point here is that oscillatory activity and synchronization have been linked with the processing for perception, motor control, and memory. So you might be asking yourself, okay, how does HIV and neuro-oscillations go together? Well, there was a study by the Wiseman lab in 2018, which was actually one of the first studies to focus on a distinction between the neuroactivity of patients with HAND and just HIV without any impairments. And this all came about because a lot of studies had recently been coming about the degradation of white matter, especially in the beginning stages of HIV-associated neurocognitive disorders. And these studies were saying that this degradation of gray matter was affecting the communication between these different neuronal groups and neuronal regions, right? And then the imaging studies that also came out with these studies showed that there were impairments in the participants and the patients' visual perceptual and visual attention networks. And so neuro-oscillations at that point have been found to organize the processing of visual stimuli in the occipital cortices and visual attention function. Kind of like what I mentioned before with neuro-oscillations, how they're usually at the top of the processing chain for perception, motor activity, et cetera. So in this study, the Wiseman laboratory had thought that oscillations could end up giving an insight into the mechanisms that actually underlie these attention deficits in HIV patients. So for sake of time, I just want to briefly go over some of the findings of this study. And I'll first start off with the theta responses. So of course, oscillatory visual theta responses were reduced in HIV infection and in hand. And you can see in this diagram, you have the controls in the green, HIV infected without impairments in the black, and hand in the red. And you can see just in this diagram, looking at the amplitude, that the HIV infected and hand were significantly decreased in comparison to the controls. And this is very important because theta band synchronization observed, especially in the posterior cortex, or posterior cortex, has really been correlated with stimulus recognition. So you can imagine if there's a deficit in that, then it goes to show that there's some correlation or some link between the visual perceptual deficits that HIV and or that hand patients are going through at the time. And so in addition to that, there was also some discoveries about alpha and gamma activity in the visual cortex. So alpha activity first, alpha desynchronization in the occipital regions has been correlated with top-down processing and inhibitory release. And so it was also interesting to see that the participants with hand exhibited significantly increased alpha activity compared to HIV and the controls. And then looking at gamma, gamma desynchronization has been linked with fine scale encoding of visual stimulus features. And so in the same manner with alpha desynchronization, it was interesting to see that both hand and HIV patients had elevated spontaneous gamma activity compared to the controls. And so lastly, just to look at the task efficiency or accuracy within this, it seems like the hands ended up decreasing, the accuracy ended up decreasing throughout the behavioral studies. And this kind of showed the correlation between disease severity and the decrease in accuracy, and almost in a stepwise manner. So all of this to say that HIV infection and hand status differentially affects neuro-oscillations and rhythmic activity in the brain, and that these neuronal responses are directly linked to behavioral and psychological performance. And so there needs to be more light shed on this area, especially with the fact that if we understand neuro-oscillations and specifically how they're tied to neuronal responses, we can use them to end up tracking the progression of hands, essentially using them as a biomarker for hands, and making more efficient, more accurate prognoses and diagnosis in the future. So here is a list of my references, and I would just like to give a very big thank you to Dr. Amanda Brown. In her laboratory, Dr. Amanda Brown has been my mentor, not only through this program, but through high school and her JHIPS program. And I'm just very appreciative of all the help, all the guidance that she's given me during this time period. Dr. Claire Carter, of course, with the professional development sessions, in this Johns Hopkins Neuroscience Scholars Program, it's not only just about, the science, it's also about your development of character. And I'm very appreciative of what Dr. Carter was able to do during that time period. Miss Heather Thomas, she's been an incredible help, always staying on top of things and just making sure that we're all okay and presenting us with these different opportunities. And then just in general, Johns Hopkins, all my fellow neuroscience scholars, you guys have been incredible this summer, and I've learned so much from you guys, and I'm excited to see what you all do in the future. Thank you.
Kirian, it's so wonderful to see the growth you know, from last summer and then taking even deeper dive this summer. So I'm so excited for the research and where you're going to take it from here. So outstanding job. Any questions for Kirian? Go ahead and unmute yourself. Okay, excellent. Let's see here, 118. We'll go ahead. Thank you, Kirian. Excellent job. Okay, we will go ahead now to Ben Lowe. Ben is keyed up and ready to go. Okay, Ben is one of our new scholars in the third year cohort who's been working with Dr. Ratnanathan. Take it away, Ben.
Okay. Can everyone see? Okay. So hello, everyone. My name is Benjamin Liu. I'm currently a advising junior at Pomona College in Claremont, California, studying computer science and neuroscience. I'm a first-year scholar with the Johns Hopkins Neuroscience Scholar Program. This summer, I had the wonderful opportunity to work with the MIM Ratnanathan Lab with the Center of Imaging Science at Johns Hopkins University. Today, I'll be talking to you all about the volumetric reconstruction of the feline auditory cortex from histology. So what is histology? Histology exactly is basically all the minute, the microscopic details. What we're mainly concerned with is the auditory cortex. So we're primarily looking at a specific section of brain histology. So we researchers at the Hanover Medical School in Germany, they collected SMI histology of auditory cortexes in cats. The data that I worked with over the summer consists of 2 individual cats with approximately 60 braces per cat. These are very big data files, so like about 1.5 to 2.5 gigabytes per image, you could say, which is very good for detail preservation. Like all the details are preserved, but it's very hard to work with directly because, I mean, everyone should probably know that it takes a long time to even download like a gigabyte file, much less work with one. So to make it easier for us to work with, we have to send these histological slices through a pre-process, pre-processing, in order to be able to work with the images easier. So this is basically done in a Jupyter notebook with Python. We have some packages like Simple IDK, where we take every single image for each cat. We put it through this process. So first we downsample it, which basically makes it a lot smaller of a file. I listed kind of the example images here. Like for the example, this one, the original was 1.66 gigabytes. Now it gets downsized to 30.3 megabytes, which is a lot smaller. Then we turn it to grayscale, which makes it easier to analyze it. And then we turn it into a mass. So once we do this for all of the images that I mentioned here, is that we kind of put them together in a collection, which will help us kind of reconstruct this auditory cortex. However, there's something very important that we have to have. We have to have a volumetric reference, which is required in order to accurately reconstruct a 3D model from a 2D histology. So the easy example of how to explain it is the banana problem. So if you sliced up a banana into like 15 or so slices, and you put them, try to tell a computer to just try to align them together without an existing model, it would likely give you something like figure E, which is obviously not what a banana looks like. So what we have to have is a 3D model like F on the side, which would help the computer figure out how it actually should look like. So using that in our work, we have used something called a catalyst, which is basically essentially like an existing like reference atlas of a cat brain MRI. So this is a key step in helping us reconstruct our auditory cortex. So next, we have to figure out the region of interest. So we're working with the catalyst. We have to find the area in the brain that we want to kind of reconstruct, because what we're working with, we're not working with the entire brain. So I work in a program called ITK Snap, and I have to do segmentation of specific images in the atlas based on the histological slices that like shown right here. So we would go through our entire collection and then segment them so we can make a mask and kind of put them together to kind of reconstruct our model. So here comes alignment. So this is kind of the results of our investigation. So the image on the left, which you see, is what we would have gotten without our catalyst like 3D volumetric reference in order to kind of show us the intended shape of it. And then obviously to the right, it's correctly kind of like formulated in the shape that like a section of a brain should look like. And basically the most important thing about this is that like these reconstructed volume can help us accurately measure like thickness of the auditory cortex and other regions of interest in maybe other studies in future works. I wanted to acknowledge the efforts of Drs. Kwame Kutin and Drs. Tilak Ramanather for their helping me learn the ropes and getting acclimated and integrated in the lab. I'm privileged and thankful for all the opportunity. Dr. Kutin has been a great mentor and is easy to work with and has always been very helpful and eager to answer questions. Also, thank you to the Johns Hopkins Neuroscience Scholar Program for giving me this opportunity to do research and also meet such wonderful people. Thank you to like Dr. Amanda Brown, Heather Thomas, Dr. Carter, everyone else involved, and of course all my fellow cohort mates and everyone else. I do appreciate a lot. I learned so much from you all, and I appreciate it a lot. And then here are my references for my study. Thank you.
Great job, Ben.
Thank you.
Especially dealing with those 1 to 5 gigabyte
Yes.
images. Okay, we've got a question from Valeria. Maybe Valeria, yeah, I think you have to come up to the stage. So
Yeah.
Okay.
No, go ahead, go on.
Oh, you can go there. You have your mic on, Heather. Okay.
Hi, Ben. So, like I found.
Oh, maybe take off your mask.
Oh, all right.
So I found that interesting, like very interesting, because it was also kind of similar to what I was doing over the summer. And I wanted to ask whether why the files were so big initially, why were they so dense, and after downsizing them, did you find any loss or like any significant loss to the files, as in volume, clarity, etc.?
The files were so big, I don't exactly know why exactly, but I'm pretty sure just like they like collected it using like their very like special like expensive instruments, so they can just make that like super detailed images. In like kind of like downsampling them, we didn't really lose much. I mean, there's just so many like details that like it's kind of obviously we lose something by downsizing it, but it's what we were more concerned about is like kind of like the general like shape of it, and like it's different, like the size of like the different like structures inside and everything. So in the context of our like research, it didn't matter too much.
All right, thank you.
Thank you for the question.
Marcus has a question.
Ben, great presentation. I was just wondering, like that was a really great detailed analysis of how you do the histology. I was just wondering, is there like, what is the more explicit purpose for it? And is there a reason like felines were chosen as a model?
For our lab, exactly like what the specific thing I was working on, we were just working with deaf cat histology. So that's basically, we were just maybe like trying to see whether there's like a difference in like the cortical like thickness of the auditory cortex between like our deaf cat histologies and like a normal cat. So that's like kind of like the specific purpose. But I believe that like this like process in general could be used to like learn more about like other structures. Like it doesn't just have to be about the auditory cortex. It doesn't just have to be about like the cat. I think you can do it with, it has a lot of more general uses too, I think.
Okay, thank you.
Thank you.
Awesome, great job in discussion. And collaboration. I'm so excited, right? You have the knowledge right here in your group. Excellent. And so next up, but not last or least, is Marcus Milani. He comes to us, and he's going to be a senior at the University of Florida. And he's going to be telling us what he's been doing in Dr. Moss's lab. Marcus.
Hello, everyone. My name is Marcus, and this summer I've been working in Dr. Moss's lab studying neural response to social behavior in the big brown bat. Bats are an excellent model for communication because they see through sound. They navigate through their environment using ultrasonic echolocation calls and use the returning echoes to navigate the environment and to forage for food. But while they're actually navigating the environment through sound, they also use social calls to communicate to one another. It's been well documented that there's a wide variety of social communication calls in many different bat species, including distress, infant isolation, and to like designate territories, to describe a few. And so returning back to echolocation, bats are able to localize objects by processing differences in the perceived arrival time, intensity, and spectrum of the returning echoes. And so the bats we investigated this summer, the big brown bat, also known as Eptepicus fuscus, utilizes a specific type of echolocation called the frequency modulated call, or FM call. The big brown bat also produces a unique social call called the frequency modulated bout. These FMB calls are used to claim food while foraging. And in prior research, it's been documented that these FMB calls have been found in both male and females. Well, actually, sorry, let me correct myself. So these FMB calls have only been documented in males. But the interesting thing is that male and female bats have been found to have FMB selective units in their inferior colliculus. And so this is a really good feature showing the structure of these FMB social calls and the FM echolocation calls. The top two portions of the figure are typical FMB calls in the big brown bat, and the bottom portion is the FM echolocation calls. And the interesting thing to note is that they have very similar frequency modulation. So they go from about 100 kilohertz to 20 kilohertz. And they also have very similar durations in terms of milliseconds, but they do have different structures. And so this actually poses a unique challenge to the inferior colliculus, which must be able to discriminate between these two types of calls that have very different meanings. And so the questions we're posing in this study is, does context modulate neural responses to communication sounds in the inferior colliculus? And more specifically, we're looking to see if there's a difference in real-time emittance versus playbacks. And is there a difference between the bat's own call versus the calls of other bats? And is there a difference within the context of a competitive foraging environment between communication and echolocation? And so the inferior colliculus, which is our object of study, is a segment of the midbrain responsible for processing auditory information. The IC can discriminate between sound frequency, and it can differentiate between social and echolocation calls. And so what I was referring to earlier about how FMB calls and FM echolocation calls are very similar in terms of frequency modulation, the IC is still able to discriminate between the two. And so our experiment investigates the selectivity of the IC to social and echolocation calls, however, in the context of a competitive foraging task. And so I think the most important part of our experimental design is the 16-channel neuronexus implant, which is surgically placed in the left side of the bat's inferior colliculus. And if you look at the figure to the right, you can see that there are electrodes of varying length which allow us to measure a variety of neural responses from different layers of the IC to give us a really robust picture of what's happening in the IC during our experiment. And so to begin training the bats, we individually place bats in a cage with a bowl of mealworms. The bats must learn to locate the bowl and using their echolocation and finish eating within a time frame of 10 to 15 minutes. And this foraging task is different from how the bats are used to being fed in the lab, which is they have typically access to mealworms anytime they want. And so when we begin our data collection, we transition to a competitive foraging task in which we take two bats, pair them together, and place them in a cage with one bowl of mealworms. And so we use a camera mounted above the cage to record their behavior and 3 microphones placed on different sides of the cage to record both their echolocation calls and their social calls. And when the implanted bat is selected for a trial, we use a Plexon recording system that's able to measure the neural response from the IC. To add another layer of complexity to our experiment, on specific days, we used an audio playback system to play the recording of the same bat's trial from the previous day. So to kind of elaborate on that, during the playback trials, the bats were allowed to forage independently, similar to their training, but while they're eating, we're playing a recording of their previous day trial. And so the purpose of these playback trials is to investigate if there's any change in behavior or any change in neural response to hearing the bat's own social and echolocation calls. And so if you take a look at this picture, this is just a great summary of our experimental design. If you look closely, you have the cage and you have 2 bats clustered in the center, which is where the mealworms are at. And if you look a little bit more closely, there's a wire connecting the plexon recording system to the implanted bat, which will allow us to see what's happening in the IC. And so while this experiment is still ongoing, we do have some preliminary results. We have successfully recorded neural responses from the IC. And if you take a look at the figure on the right, we're using MATLAB to isolate different waveforms recorded by each electrode channel. And the way we analyze these different waveforms is by looking at the waveform shape and interspike interval. And if it's lower than 10%, we compare it to the average and we classify it as a unit. And so the audio data is being processed using Avasoft, which allows for echolocation calls and social calls to be identified and timestamped. And lastly, the camera recording is analyzed using JWatcher, which allows for specific behavior like eating, echolocating, cuddling, among others, to be timestamped. And so once we finish our data collection, we're going to have this huge mountain of data, which we'll process through. And what it'll allow us to do is to overlap all these recordings. And so we're able to see specific moments. And hopefully that'll give us a very holistic and clear picture of how does the context of a competitive foraging event change both behavior and also change the neural response. So I'd really like to thank my mentors at the Bat Lab, Angie and Dr. Moss. And I'd also love to thank Dr. Brown, Heather, Dr. Carter, Dr. Ratnanther for really allowing this program to happen this summer. Thank you so much.
That was outstanding. Wow, such a deep dive and so clear, right? Because, you know, the secret is I'm learning with you guys.
Yeah.
And yeah, wow. Some questions from the audience? Go ahead. Oh, we have from Valeria. Oh, you can go first.
So how often would the bats do training? Also, how did you avoid the bats getting overweight from like eating?
So we train the bats every day except one day a week, typically Sunday. And we also weigh them every day. So we're able to monitor, you know, how many mealworms they get a day. So typically anywhere from 10 to 20. And so our healthy weight range is about 12 grams on the lowest and then 20 grams on the highest. And so we try to keep them in between.
So are they flying around? Because there's this challenge. The mealworms are there.
Yes.
But I guess the box is a certain size. Perhaps it doesn't allow them to fly around.
Yeah, so they're not flying around. They're all on the ground and interacting on the ground.
Okay, because the echo is for positioning.
It's positioning, yes.
Oh, so is that a limitation then?
I wouldn't say it's a limitation because they still do echolocate even while they're not flying. So they're constantly using it to give a 3D picture of the environment, regardless of if they're flying or they're crawling.
Okay, I didn't know that. Awesome, cool.
Yeah.
Some other questions. Let's see here. Great work, very clear. Okay, great. Amazing work from Jennifer.
Thank you.
Okay, wow. What a wonderful high note to end on. So let's see how we're going to do this next part. Because I have a little something for you. Those that aren't here, we will send it to you. And so maybe what I'll do is I'm going to go, you can see us here. So maybe I'll turn on the volume over there. Just bear with us. Turn on the microphone here. Let's just go here full screen. And let's see here, speaker. Okay, everybody in the audience can see? Okay, great, thanks, Ben. Okay, what I'd like to do, I have a little gift. And I know this is not the end, it's just the beginning of a lifetime of learning, of science, of relationship building. And so our first comment on Elizabeth. Let me just go get the gifts here. I can bring this over. Oh, thank you.
I can,
grab cards here.
we need to see everybody. Go ahead and I'll turn it here.
Okay.
Go ahead and stand up. Elizabeth.
You know,
Come on up.
this journey occurred when you applied. And Heather and I, we read your applications and we have a rubric. And we have a rubric that George Shreef was so instrumental in designing, which has just worked fantastic in a fantastic manner. And it is just so amazing for us how each individual brings something dynamic. And when I spoke with Elizabeth, I was like, well, I'm not sure, but she's like, well, you know, women are underrepresented. And we know that people, you know, from Asian descent, as they go higher and higher, sometimes, right, there are barriers. And so that, I was like, you're right, you're in. And it's been phenomenal. Just the discovery, you see, based on our Clifton Strengths Finder that we've done. You know, I'm a learner, I'm an achiever, but I need harmony. But I just have a passion for learning. And Elizabeth discovered our research, dived deep and found link to the lab that she's currently in, which specializes in RNA splicing. And so it's just phenomenal. She's taken a deep dive and we're just on a fantastic pathway. And I'm just so delighted to have you and to have met you and that you chose our program. And so I'm just looking forward. So this is a little something for you. Oh, you can grab any color. So.
Thank you so much.
Thank you.
Next, we have Ariel Timothy. I spoke with her a little bit, was that last week? And again, I've known her since she was a high school student and I've been working on her. She's my real experiment. She didn't know that, but I'm confiding in her and I think she's understanding because there's this struggle and she has to realize her awesomeness. When I saw her yesterday, I could see the future Dr. Idowu. And so just keep focused and you are going to get there. And I'm so happy to be on this journey with you.
Thank you, Dr. Brown.
You're welcome. Next, we have Rose. Rose, again, reading your application, it was phenomenal. We're like, wow, our students are so diverse. They're bringing so much to our program. We're just so excited and delighted to have you. Dr. Carter, in terms of, you know, the professional development topics were spot on because our scholars have shown this summer resilience, persistence, and continue to believe in themselves, right? And talk positively to themselves. And so I'm so delighted and proud of what you've accomplished so far this year, and we look forward to the next 2 years. Thank you, Rose. Okay, next up we have Marcus. This has just been phenomenal. I heard somebody said, oh, he's so tall. And I had no idea because I had no idea you were this tall. It's wonderful.
It's hiding behind Zoom.
It's hiding behind Zoom, yes. And it's just, again, this, you know, Dr. Tilak, Dr. Ratnanathan, what we're doing, inclusion and being mindful. You know, it's Tilak who's been fighting for these captions and fighting for, you know, changing our institution. And he's the one that did it because there was no captioning and kept going forward. So that speaks to all of our deaf, hard of hearing scholars. Has been outstanding, but not only you've seen the outstanding science and what is capable. And so we're just so excited. Thank you so much for your excellence.
Thank you.
Just look forward to the next steps. And thank you. Thank you for, you know. being able to come to Baltimore in person. And the rest of you, we're looking forward. So you're not gonna miss out. You're gonna get all of this and more when we see you. Okay, and next we have Valeria. Valeria, it's just been outstanding and in a short time. She has just shown her massive leadership qualities. She wants to know Baltimore. She has walked over around so many places in Baltimore. She has embraced what it means to be a member of the Johns Hopkins community. She's living it, and she's becoming also as well awesome scientist. And so I'm just so excited and delighted to have you in the program. And thank you for your leadership and everything that you've done this summer. And I'm sure you will. Okay, and I have some other cards here, which is going to go ahead and make the announcement.
You want to sit back down?
Oh, okay. Oh, this is nice. So even though you're not here, right? So I have cards for all those that were here, but I have my list. Let me go. Let me make sure. Okay, Darius. Awesome job. I think I said it before you presented, because I know you've been waiting for this experience for a couple of years. You got it. You seized it. Keep up the good work. So delighted to have you in the program.
Thank you.
Jada, I met Dr. Kamkwala in the hallway earlier this week. She said you're doing an outstanding job. She's extremely delighted, and I'm so glad that you brought your excellence and motivation to the task. So keep up the good work. Look forward to seeing you. Excellent job. Kirian, you also exemplify persistence, intensity. You guys don't know, Kirian has like all A's. Maybe some of you others of you do too. I was like, send me your transcript. All A's. It's like, wow. But he's very modest, he's confident, and Kirian, we're gonna find the right place. But you've done the preparation. Keep up the great work. Is phenomenal. Excellent job. Is Catalina here? Was she able to attend? Anybody see Catalina? Okay, we'll just, because I want to get to those that are here. And Princess is probably not here. I don't think, was she on there? Okay, Ashley, phenomenal job. I'm just so excited to see you continue to develop as a scientist. And I'm looking forward to having you in the lab so you can actually test some of your hypotheses, but you're doing an outstanding job. Ashley originally started in neurosurgery, and then she's come to HIV, and I'm just so excited to be a benefactor of her interests. Excellent job. Thank you.
Yeah.
Riley, is Riley with us today?
Yes.
Okay, awesome. Riley, again, outstanding job. You are from our alumni cohort. You had an amazing experience with Dr. Bush and their whole team. We saw from your presentation yesterday that you went deep and you really found your passion to be a future advocate, or you're an advocate now for young people because you remember what that was like. And so proud of your accomplishments. Keep up the good work.
Thank you.
Zandy's probably taking a break, right? Is Zandy on?
Sandy couldn't make it.
No, Zandy, man, she just goes. She's high energy, high intensity. She needs a break. I think Erica's probably visiting with family. Is Erica on? Oh, Erica's there. Okay. Erica, phew, you're another one, high intensity. And we're gonna see you in, I guess maybe another week or so. Thank you so much for your leadership, your advocacy. You just jumped in. You're like, oh, can we mentor the high school students? Can we, initiate journal club or help with the journal club? It's just been phenomenal how you have inspired your fellow scholars and on the science side. And you've also been resilient because it has taken some time to find a mentor. But again, you must be a learner achiever. You love to learn and you love science. And I'm just so delighted to have you in the program. So thank you.
Thank you.
And we have Brandon. Is Brandon on?
Yeah, I'm here, Dr. Brown.
Oh, I couldn't hear. You are there? Brandon is
Yes.
here. Oh, excellent. Brandon, I think I mentioned it yesterday, and I have in a couple of our sessions, the area and the passion that you have for drugs of abuse and the research is just phenomenal. Looking forward to having you here to actually do the experiments. So hang in there. I know it's gonna be fine, and you're doing an outstanding job. And look forward to having you next summer. So keep up the good work.
Thank you, Dr. Brown.
Was Jacqueline able to attend today? Is she there, Ben? Because I can see you. Is Jacqueline on the call? Not today.
No.
Okay, thank you. And then Jennifer. Wow, Jennifer is a heavyweight. I mean, you could just imagine, we will never forget, right? The first Heather and I, right? Jennifer, actually it was the team, Jennifer, Riley, and Morrissey, Olivia. And again, strong leadership skills, persistence, advocacy, making us better, making our program better. And then now with the science, she's been able, you see, to pull it together in a beautiful and awesome way. Jennifer, I'm just so excited for your next steps. And so congratulations on a great job.
Thank you so much, Dr. Brown.
Shaheen, wow, I think we've also said it many times as well. And so we're just so delighted to have you on board, that the match between you and Dr. Wu and the group, it seems like a perfect match. And so, your own story is compelling. And so it's just going to be really amazing to how you add to our Hopkins community and what you will do for others. And the story that you'll be able to tell about your journey is pretty awesome already. And it would just be more awesome in the future. Thank you so much for your excellence.
Thank you, Dr. Brown. You're amazing.
Okay, and was that everyone? Did I get everyone? I think so. Doctor, oh, is somebody saying something? Maybe you can put your sound. Oh, let's see. I have the sound here. There we go. Let's see. Here. Okay. Oh, Ben, the last for the best, the best for the last. It's been phenomenal because again, you've pushed us, and again, you're coming. You found us from all the way from Pomona, right? University of Pomona or Pomona College.
Pomona College.
In California, yeah, and there was maybe, well, I guess it was through as well, connected with Dr. Ratnanathan. But you are breaking barriers. And I really liked your presentation today. Was very clear and gave us greater insight. And you even seemed to understand the impact based on with the discussion in the discussion session. And so that's really phenomenal. And so it seems like we've got a good alignment here. And so I'm excited for your future and what you're going to do in the field and for others. So thank you for your patience, and we look forward to seeing you in person next summer. Great job.
Thank you.
You're welcome. Okay, did I get everybody now? At least all of the scholars. Okay, and so next, I mean, you know how we had to pivot this year. We all have had to pivot. And so our team worked so hard, and you guys know that. And so the pillar, and it's like my right hand, is Heather Thomas. And so I have a gift for Heather. That would go nicely in your nice desk area with the beautiful light.
Well, I love being your right hand. I really do.
So
Thank
couldn't
you so
have done
much.
it without you. And it's just a great partnership and a passion for both of us. So I think everybody understands that.
It
And
is.
hope that it can make it continue.
Absolutely. Thank you all so much. This is my heart. This is where my heart is. I finally found my path. What I want to do for my career. It took me to my 40s, but I just absolutely love education, and especially research education. So I am your biggest cheerleader. I think about you guys all the time, and I just want the best for you, for your future. And you are super high achieving scholars, and I'm just looking forward to your future. All right, and I'm happy to know you. You're gonna be famous. Thank you all so
Awesome,
much.
thank you. And Dr. Carter, are you online? Are you there? Is Dr. Carter there still?
Yes.
Oh,
I'm here.
go ahead. You were asking. Dr. Carter
I am here.
started this journey years ago. Did you imagine we would be here? That you in retired, so-called retirement, so hard. Dr. Carter is the one behind professional development. What we do, making sure that I'm in the time. And so we worked incredibly hard. We had both of the program, and it was just a phenomenal. And so this bouquet of flowers is just a small token to say thank you and just how much you're loved and valued. And so I just appreciate, I could not do this on my own. It's my lesson.
You know, I can't believe that I joined this team. It's been 11 years now. I thought it was going to be 3 months, 11 years ago, and here I am. And I have gained many, many new young friends. All of you are my friends. You know, I talk about the family. But I'm most appreciative to cohort 3 coming in at the short notice when we weren't even sure, and you connected right away. And that was excellent. And then to cohort 1 and 2 who reached back and became peer mentors. I mean, they didn't let anything fall apart. They were there for you. So that talks about the community that we have. Unfortunately, I didn't hear all the presentations. I was with a Hopkins doctor for 3 hours yesterday, making sure I was in good health. I wasn't sure if I was going to make it through the summer, but I'm good. Thanks to Hopkins, I'm in good shape. So I did hear the 3 young men at the end. And those 3 guys know I always talk about them. And you did outstanding presentations. But there's another special guy. See, I'm sorry, girl. I'm hooked to the guys. But that's all good. And there's my musician at the end. And I hope you stay with the family, and next year that you will be able to serenade us and give us the music. Because we always say Thursday without music is not an option. That's our motto. So I wish you well. My guy with the deep voice, he's awesome. He's always on task. He's going to make it. I know he's going to make it. And Ben, I love your smile. And Riley and all the girls, Sarah, all of them are so very special to me. And I hope we stay in touch. Don't be surprised if you get notes from me. And hold on to your rock, because that's how we move, okay? Best of luck to you all.
And Tila, Ratnanka already described the rock that brought everything together. So thank you for your partnership. Would you like to close us out?
Well, I really like to thank Dr. Brown and also especially Heather for taking on a lot of work administratively. So Heather should be recognized for her hard work. And also we're trying to coordinate everything over the pandemic. And some people managed to come to Hopkins. I managed to see one of them. And hopefully next summer we'll be all together and we can do more things together. And but most important, we have learned how to do science and also how to become leader and also how to aim for the moon.
Moon, yes. Thank you. So enjoy the rest of your summer. Take a break. And yeah, you know we're always available. And so yeah, thank you. I don't have to say too much because
I know.
Oh,
I
go
don't have anything, but I wish I would have gotten these flowers for Amanda because you know she's the leader. She is our leader, you know, and I just thank her so much. She's put these grants together. She's written these grants. She's gotten them funded, and that's not an easy task at all. But as you can see, we've developed an excellent several programs with the high school program as well. But we are a family, and you know, thank you so much for always caring for us so much from the bottom of your heart. I know you care for all of us, and I care for you.
Don't make me cry
As
now.
we all do. So thank you so much.
You know why? You know the
Dr.
secret? It's because I'm here, because you're here, because I've been there, right? And so I do this so that it will be a little easier for you.
Yeah.
It's all good.
See? Yeah. That's beautiful. Dr. Brown.
Bye. See you soon. And we'll get your gifts to you. We'll send them out to you.
Bye, everyone.